across all species, we actually found that the age-dependent model was the better fitting. Also, just so if you don't know, lower AIC is a better fit. So we found that the age-dependent model was a better explanation for neonatal survival, which is great, because then we can delve a little bit deeper, because we also wanted to ask whether we wanted to analyse the relative frequencies of these positive and negative maternal effect senescences that we saw in the effect sizes with a simple sign test. And we were essentially asking whether maternal effect senescence was widespread. And the null hypothesis was essentially there is an equal chance of finding positive and negative maternal effect senescence. And when we looked at it across all the 32 species, we actually found that there was an equal chance of finding positive and negative maternal effect senescence. And then I broke it down into natural populations and lab-based populations, and natural populations with conspicuous maternal care. And we also couldn't reject this null hypothesis. So there was equal chance of positive and negative senescence. However, when we looked at lab-based species without this post-hatching care, we actually found that we could reject this null hypothesis, because in the lab they actually showed there was a strong evidence for maternal effect senescence, essentially. So when we did see this maternal effect senescence, we then wanted to ask which evolution model better fit the observed pattern. And running for all the species that showed maternal effect senescence, we actually showed that the original model was a better fit. So the age distribution of females was a better fit. But when I looked into it a little bit more, I removed any laboratory species and only included those that actually were natural populations with conspicuous maternal care. And we actually found that those natural populations where maternal effect senescence was occurring, we actually found that it actually favoured the age distribution of mothers rather than the age distribution of females. So we can kind of say that in total it was favouring the female age distribution, but when we actually look at more natural populations, they were focusing on it actually was better explained by the age distribution of mothers. And one reason for this could be actually because the lab populations showed demography that is quite artificial, so it may not be actually appropriate for this analysis. So in summary, we kind of showed with this analysis, because it's ongoing, it's going to improve and it's going to change. We showed so far that there's limited evidence for maternal effect senescence. However, there is strong evidence for it in the lab. And when it does occur, it's better explained by age distribution of mothers when we're looking at natural populations. So the new model is actually a better fit when we're looking at natural populations with conspicuous maternal care. But female age distribution is actually a better explanation for laboratory populations when we're looking at laboratory population maternal effect senescence. So the next steps that I'm going to be doing with this work is essentially I'm going to be widening my sample size and trying to look at more natural populations to see if the age distribution of mothers is still a good fit. I'm also going to solicit raw data from investigators, and I know I've asked a couple of people already at this conference. One thing I didn't do in the previous analysis, I didn't correct for phylogenies, and this is something that I'm looking to do in the coming future analyses. And lastly, the other thing I was going to do was include other life history traits. So we looked at post-hatching care, so presence or absence of conspicuous maternal care, post-hatching care. And so I'm also going to look at other life history traits, such as maximum longevity and body size, and see if this changes the fit of the evolution models. So with that, I'd like to thank my funding bodies, EastBio, BBSLC, Jacob Morad for his assistance, and the University of Edinburgh. Thanks.
So demographers probably have the richest data set with humans, so why did you exclude them?
Just because they kind of represented, they were too complex, essentially. We wanted just to use species where they didn't have the complex social system in place. So, that answers your question.
So maybe you said this and I missed it, but what were the 35 species? Were they all mammals?
No, I'm sorry, did I explain that? Okay.
I might have missed it.
So it was 35 papers, there were 32 species, and it was kind of a half and half split between, there was about 14 mammal species, ranging from cheetahs, jaguars, seals, soy sheep. There was also a whole host of other invertebrates and bird species as well. So invertebrates were Drosophila littoralis, stem boll weevils. I can't remember all of them off the top of my head, but there was quite a mixture.
Not having seen the distribution of the data, it seems likely that the lab wild comparison is heavily confounded by body size and phylogenetic effects, thinking that the lab species are more biased towards invertebrates and
Yeah.
smaller vertebrates. Also, the lab populations are likely to have females reproducing later in life
Yeah.
than field populations because of natural mortality. So I guess it's just sort of a caution about attributing differences between lab and field to lab versus field rather than.
That's definitely something we've noted, yeah. Thanks.
Along those lines, do you think there's a possible genetic influence with lab populations versus natural populations? A lot of lab populations are very inbred,
Yeah.
so very low genetic diversity.
I don't know. Do I have enough time to answer?
Yeah, this transitions.
Okay.